Nearly 11 kilometers down, at the bottom of a crescent-shaped scar in the Pacific seafloor called the Challenger Deep, the pressure is more than 1,000 times what you're feeling right now at sea level, equivalent to roughly the weight of 50 jumbo jets pressing down on a single square meter. The water sits just above freezing. No sunlight has ever reached this deep, not once, not for the entire history of the planet. By every reasonable expectation, nothing should be able to survive down there. Something is thriving down there instead. Over the past 2 years specifically, a wave of expeditions has returned from the Mariana Trench with results that have genuinely surprised the scientists conducting them. Thousands of new microbial species, the deepest dense colony of large animals ever documented, and biochemistry that doesn't behave the way researchers assumed it would. This is what they actually found at the deepest place on Earth, and why it's forcing a real rethink of what extreme environment actually means. Before getting into the recent findings, it's worth sitting with the scale of what we're actually talking about. The Mariana Trench is a roughly 2,050-kilometer-long depression in the Pacific Ocean floor, formed where the Pacific tectonic plate is being forced underneath the smaller Mariana plate in a process called subduction. Its deepest point, the Challenger Deep, plunges to approximately 10,184 meters below sea level, making it the deepest known point anywhere on Earth's surface. For comparison, if Mount Everest were somehow dropped into the Challenger Deep, its summit would still sit more than 2 kilometers below the surface of the ocean. The trench was first identified in 1875, during the pioneering British Royal Navy oceanographic expedition aboard HMS Challenger, the survey that eventually gave the deepest point its name. But identifying it and actually reaching it turned out to be two very different challenges, separated by the better part of a century. It wasn't until 1960 that anyone actually descended to the bottom when Navy officer Don Walsh and Swiss oceanographer Jacques Piccard piloted the bathyscaphe Trieste down to the Challenger Deep floor, a feat that wouldn't be repeated by another human being for 52 years, until filmmaker James Cameron made a solo descent in 2012. Since then, a small handful of additional expeditions, including several dives by explorer Victor Vescovo and his team starting in 2019, have added to what remains an extraordinarily short list of people who have ever actually visited the deepest point on the planet, somewhere in the range of 24 individuals total, fewer people than have walked on the moon. That scarcity of direct human visits is part of why the trench has remained so poorly understood for so long, and it's exactly what's changed over the past couple of years. Not through more human dives, but through a combination of advanced remotely operated vehicles, uncrewed submersibles, and considerably more sophisticated sampling and genetic sequencing technology than anything available even a decade ago. The single largest recent finding came out of a project called MIR, a China-led research effort involving Shanghai Jiao Tong University, working alongside international collaborators, including the University of Copenhagen and Lawrence Berkeley National Laboratory. Using the crude Chinese deep-sea submersible Fendozhe, meaning striver, the team conducted an extensive survey of microbial life across the Hadal zone, the term oceanographers use for any part of the ocean deeper than 6,000 meters, sampling sediment from some of the deepest trenches on the planet, including the Mariana Trench itself. Published in the journal Cell in early 2025, the results were genuinely striking in scale. Researchers identified roughly 7,000 distinct microbial species living in these deep Hadal sediments, and of those, an extraordinary 89. 4% had never been documented anywhere before, meaning the overwhelming majority of what the team found was, quite simply, new to science. The study's analysis went beyond just counting new species. It also examined the underlying evolutionary pressures shaping this community, finding that the extreme conditions at these depths appear to favor two distinct survival strategies among the microbes living there. Some species evolving toward highly streamlined specialized adaptations suited to one narrow set of conditions, others evolving toward broader, more generalist versatility capable of tolerating a wider range of variable conditions, with both strategies actively competing and coexisting, rather than one simply dominating the other. Researchers involved in the work have specifically framed these findings in terms of broader scientific value beyond simple novelty, noting that this level of genetic and biochemical diversity represents a genuinely untapped resource, potentially containing entirely new genes, protein structures, and biological functions that could eventually inform new drug development or other biotechnology applications, precisely because these organisms have evolved biochemical solutions to survive conditions, crushing pressure, near-freezing temperatures, complete absence of sunlight that terrestrial life has never had to contend with. Microbes surviving at extreme depth is one thing. Microorganisms are famously tenacious and have been found thriving in a wide range of environments most other life can't tolerate. Considerably more surprising was a separate 2025 finding, published in the journal Nature, documenting something researchers hadn't previously expected to find at this depth at all: a dense, thriving colony of large multicellular animals. Using the same Fenduge submersible, researchers conducted 23 separate dives, specifically targeting the Mariana Trench, and identified what they described as the deepest large animal colony ever directly observed. A dense aggregation of tube worms and mollusks, including thick beds of clam-like bivalves, researchers nicknamed the clam bed, spread across a startling depth range, from roughly 8,100 feet down to nearly 31,000 feet, close to 6 miles beneath the surface. That's a genuinely enormous vertical range for a single interconnected biological community to occupy, and it pushed the known depth limit for this kind of large animal colony considerably further than researchers had previously documented anywhere on the planet. What makes this discovery scientifically significant isn't just the depth record itself, it's what's actually fueling this community. Almost all life on Earth's surface ultimately depends, directly or indirectly, on energy originally captured from sunlight through photosynthesis. But no sunlight reaches anywhere near these depths. Instead, researchers found the community was being sustained through chemosynthesis, a biological process in which microorganisms generate usable energy by chemically processing compounds like methane and hydrogen sulfide, rather than capturing sunlight, essentially building an entire food web from chemistry alone rather than from solar energy. The research team specifically found compelling evidence that methane was being actively produced by microbial activity at the site, with the tube worm colonies clustering densely around microbial mats that researchers described as resembling drifting snow, a direct visual sign of the chemosynthetic process actively fueling the surrounding ecosystem. The broader implication the research team drew from this finding is genuinely significant for how scientists think about the deep ocean generally, since the specific geological and chemical conditions enabling this kind of chemosynthesis-based community aren't unique to this one particular site. Similar cold seeps and methane-rich zones exist in other deep ocean trenches around the world. The researchers suggested these kinds of thriving chemosynthesis-powered communities might actually be considerably more widespread throughout the world's Hadal trenches than previously assumed, rather than representing some kind of rare, isolated anomaly specific to this one location in the Mariana Trench. Alongside these large-scale ecosystem discoveries, a more specific technical finding out of the same broader wave of Hadal research quietly overturned an assumption that had stood largely unchallenged in deep-sea biology for years. Researchers had long understood that organisms living at extreme ocean depths need some biochemical mechanism to keep their proteins from literally being crushed and destabilized by the enormous surrounding pressure, since pressure at these depths is genuinely capable of disrupting the delicate molecular structure that proteins need to function correctly. The prevailing assumption, based on research conducted at moderate depths, was that a specific compound called trimethylamine N-oxide, TMAO for short, which is known to help stabilize protein structure under pressure, would simply keep increasing in concentration within fish tissue as depth, and therefore pressure, increased essentially, a straightforward linear relationship between how deep a fish lives and how much of this protective compound its body needs to produce. The 2025 research specifically tested that assumption directly against fish collected from below 6,000 meters, genuinely extreme Hadal depths that previous TMAO research simply hadn't been able to sample from directly, and found no significant further increase in TMAO concentration at these depths compared to somewhat shallower samples, contradicting the straightforward linear increase model that had previously been assumed to hold. That finding suggests the biochemistry fish actually used to survive genuinely extreme pressure beyond a certain depth threshold may involve additional or entirely different protective mechanisms that researchers hadn't previously identified or accounted for, a reminder that even a reasonably well-established piece of deep-sea biology can turn out to be incomplete once genuinely extreme depth samples finally become available for direct testing, something that simply wasn't possible until recent advances in deep-sea sampling technology made collecting specimens from this specific depth range practically feasible. Not every significant recent discovery in this region has come from the absolute bottom of the trench itself. In May 2025, the research vessel EV Nautilus conducted an extensive expedition across the broader Mariana Trench Marine National Monument, a U.S. protected area covering more than 246,000 square kilometers of submerged lands and waters surrounding the Mariana Archipelago, spending 115 hours diving across one of the most geologically active stretches of seafloor on the planet, a region containing more than 60 submarine volcanoes, 21 of which remain actively venting hydrothermal fluid. Among the expedition's most significant findings were 2 entirely unexplored mesophotic coral ecosystems, deep-water coral communities living at depths still reached by limited sunlight, discovered along the western Mariana Ridge, neither site having ever been formally surveyed by any previous research expedition. Given that the overwhelming majority of this vast protected monument remains, In the researchers' own assessment, poorly understood despite decades of intermittent study, findings like these newly documented coral ecosystems underscore just how much of even a formally designated, actively protected marine region can still contain genuinely undocumented biological communities, discovered essentially by chance during a single, relatively short expedition. Specifically, because so little of the surrounding seafloor has ever been comprehensively surveyed before, not every recent finding at the bottom of the Mariana Trench has been a triumphant scientific discovery. In an expedition completed in early 2025, oceanographer Dawn Wright, who became the first black person to reach the Challenger Deep during her dive, documented something considerably less encouraging sitting on the ocean floor at the deepest point on the planet, a discarded beer bottle. It's a small, almost darkly comic detail, but it carries real weight precisely because of where it was found. The Challenger Deep sits roughly 11 kilometers beneath the surface in one of the most physically remote, difficult to access locations on the entire planet, a place fewer than 30 human beings have ever personally visited. Finding ordinary human litter sitting at the absolute bottom of that specific location is a fairly stark, immediate illustration of just how thoroughly human waste has managed to reach even the parts of Earth that remain, in almost every other respect, essentially unexplored and inaccessible to us. Researchers studying ocean pollution have used this kind of finding, alongside broader documented evidence of plastic and other human debris recovered from Hadal trenches worldwide, to argue that there is genuinely no remaining part of the ocean, no matter how deep or remote, that has been fully insulated from the reach of human activity and waste. Beyond the cold, seep, and methane-driven communities described above, the broader Mariana Trench region hosts a genuinely active volcanic environment that contributes its own distinct chemical energy source to the deep ecosystems living there, and understanding that system helps explain why this specific stretch of ocean floor has turned out to be so unexpectedly rich in life. The more than 60 submarine volcanoes scattered across the wider Mariana region, 21 of them actively venting hydrothermal fluid, according to the 2025 Nautilus survey, function as chemical factories in their own right, similar in basic mechanism to hydrothermal vent systems, documented elsewhere around the world, including the well-known black smoker vents found along other stretches of ocean ridge. Seawater seeps down through cracks in the volcanically active seafloor, gets superheated by proximity to magma below, and picks up a rich load of dissolved minerals and chemical compounds along the way, before venting back out into the surrounding water, creating localized plumes of mineral-rich, chemically energetic fluid that specialized microorganisms can directly metabolize for energy, entirely independent of both sunlight and the separate methane-based chemosynthesis process documented at the trench's deeper cold seep sites. That means the Mariana region actually hosts at least two functionally distinct chemical energy pathways, supporting deep sea life simultaneously: hydrothermal vent chemistry tied to active volcanic venting, and methane and hydrogen sulfide-driven chemosynthesis tied to cold seep sites like the one hosting the newly documented tube worm and clam colonies. Having both systems actively operating within a relatively concentrated geographic area is part of why researchers have found this specific region so productive for documenting new extreme life adaptations, since it effectively offers two separate naturally occurring laboratories for studying how life can be chemically fueled in the total absence of sunlight, sitting within reach of the same broader research expeditions. It's worth spending a moment on just how difficult the underlying engineering challenge actually is, since it explains a lot about why these discoveries are concentrated so heavily within just the past couple of years, rather than being spread evenly across decades of steady, incremental progress. At nearly 11,000 meters depth, the surrounding water pressure exceeds 1,100 times atmospheric pressure at sea level, a force capable of crushing most conventional vehicle hulls instantly if not properly engineered around. Vehicles designed to operate at these depths, whether crewed submersibles like Fendoge or uncrewed, remotely operated and autonomous vehicles, require pressure hulls built from specialized materials, often titanium alloys or advanced composite materials, machined and welded to extraordinarily precise tolerances, since even a small structural flaw becomes catastrophically dangerous under this level of sustained pressure. Beyond the hull itself, every single sensor, camera, sampling arm, and communication system carried aboard has to be specifically pressure rated for full ocean depth operation, a genuinely narrow, specialized engineering field that only a small number of research institutions and companies worldwide currently have the accumulated expertise to work in reliably. That narrow technical bottleneck is precisely why expeditions like the 23-dive Mir campaign represent such a meaningful shift in research capability. Earlier full-depth expeditions, going back to the original 1960 Trieste dive, generally involved a single vehicle capable of, at best, 1 or 2 dives total, often requiring extensive rebuilding or repair between each descent given the extreme stress the pressure hull and components experience on every single trip to depth and back. Having a vehicle reliable enough to complete 23 separate dives, specifically targeting the trench within a single sustained research campaign, reflects a genuinely significant maturation in deep-sea engineering reliability, and it's precisely that kind of repeated, systematic access. Rather than the occasional single dramatic dive that characterized earlier decades of Hadal exploration, that's made this recent wave of comprehensive biological survey work practically possible for the first time. Taken together, this recent run of findings, thousands of new microbial species, an unexpectedly deep and dense large animal colony fueled by chemosynthesis rather than sunlight, a biochemical assumption about pressure adaptation that didn't hold up under direct testing, and newly discovered coral ecosystems in a region assumed to already be reasonably well surveyed, points toward a fairly consistent underlying pattern. The deep ocean, and the Mariana Trench specifically, has been far less thoroughly explored than most people, including many scientists working in adjacent fields, had generally assumed. Part of that gap comes down to simple technical difficulty. Building a vehicle, whether crewed or remotely operated, capable of reliably withstanding pressure at nearly 11,000 meters depth, while still carrying the sensitive scientific instrumentation needed to actually collect useful samples and data, remains a genuinely difficult engineering challenge, one that's only become practically feasible at any meaningful scale within roughly the past 15 years. The Fenduzi submersible behind several of the discoveries described here is itself a relatively recent piece of Chinese deep-sea engineering, and its availability for repeated extensive scientific missions, 23 separate dives in a single documented campaign, represents a genuinely different scale of sustained deep-sea research access than what was available to earlier expeditions, which more typically involved a single, carefully planned dive rather than repeated, systematic survey work across an extended research campaign. There's also a more basic numbers problem. Human ocean exploration effort, funding, and attention has, for entirely understandable practical reasons, concentrated overwhelmingly on the shallower, more immediately accessible parts of the ocean, coastal waters, continental shelves, the upper few hundred meters where most commercial fishing, shipping, and coastal research already takes place. The Hadal zone, by contrast, is difficult to reach, expensive to study, and offers essentially no direct commercial incentive driving sustained exploration investment, meaning it's remained comparatively understudied for reasons that have much more to do with practical accessibility and funding priorities than any lack of genuine scientific interest in what might actually be down there. The practical upshot of this recent wave of research is that the Mariana Trench and Hadal ocean environments more broadly are increasingly being recognized as genuinely significant rather than simply curious extreme environment footnotes. The Mir research team specifically framed their findings in terms of the trench's ecological importance at a planetary scale, describing Hadal environments as playing what they characterized as irreplaceable roles in broader global biological and chemical processes, precisely because of the unique, still poorly understood extreme life processes occurring there, processes that may have genuine relevance well beyond pure academic curiosity about deep-sea biology. That framing connects directly to a broader theme running through a lot of recent extreme environment research more generally, whether that's genes and biochemistry recovered from Hadal trench microbes, or research into deep-sea giants elsewhere in the ocean. Extreme environments consistently turn out to be sources of genuinely novel biological solutions, evolutionary adaptations shaped by conditions terrestrial life has simply never had to contend with. And those novel solutions carry real potential value for fields well beyond oceanography itself, including medicine, biotechnology, and even, as some researchers involved in this specific research have directly noted, insights potentially relevant to understanding how life might be supported during long-duration human space travel, where astronauts similarly face extreme, tightly constrained environmental conditions quite different from anything found at Earth's surface. 11 kilometers down, in permanent darkness under crushing pressure and water just above freezing, life isn't just barely surviving. Based on what these recent expeditions have actually found, in at least some specific locations, it's genuinely thriving, and doing so through biochemistry and ecological strategies that scientists are still, even now, in the process of properly understanding for the very first time. Fewer than 30 people have ever seen the bottom of the Challenger Deep with their own eyes. Based on how much has been found there in just the past 2 years alone, that number and our understanding of what's actually down there both seem very likely to keep growing. There's a particular kind of humility built into stories like this one, worth sitting with for a moment before wrapping up. This is the deepest, most physically extreme point on the entire surface of our own planet, not some distant world light years away, and it took until 1960 for a single human being to even reach it once, and until the past couple of years, for anyone to actually study it with anything approaching real thoroughness. We've mapped the surface of Mars and the far side of the Moon in considerably more comprehensive detail than we'd mapped the biology living at the bottom of our own oceans until remarkably recently. That gap is closing quickly now, driven by genuinely impressive advances in deep sea engineering and genetic sequencing technology. But it's worth remembering just how large that gap still was, right here on Earth, until very, very recently.